Hello everyone, today we will be discussing the origins of life, specifically tackling three main questions. 1. How molecular interactions under early Earth conditions could have led to the formation of ancestral cells. 2. What properties of a biological molecule would allow it to be the first molecule of heredity in the prebiotic world? And 3. What evidence do we have to support the scientific theory that life on Earth had a common ancestor? So to begin, what are the possible hypotheses for the formations of ancestral cells? There are two main ideas. First is panspermia, which explores whether life came to Earth from other planets. This would include the possibility of meteors bringing low-level life forms to Earth. However, this begs the question of where and how these life forms originated. The second is abiogenesis, which suggests that life occurred spontaneously here on Earth. In this theory, it is believed life originated from simple atoms and molecules such as carbon and hydrogen, combining at high temperatures in the presence of water and lightning, and any other early Earth conditions. This transition from atoms to living cells may have taken millions or even billions of years. To test this theory, experimenters Miller and Urey conducted experiments in 1953 in which they placed hydrogen, water, methane, and ammonia all together with high-power electrodes necessary to provide reaction energy. In early Earth, this energy could have been provided by lightning. What Miller and Urey found was that simple organic molecules such as urea, organic acids, sugars, and amino acids were formed. This showed that complex molecules could have been formed under a reducing atmosphere. For life to continue on Earth after the formation of cell precursors, some mechanism had to form as a way to transfer hereditary information between generations of organisms. Due to the conditions of the early Earth, this precursor had to be capable of two main functions: self-replication and reaction catalysis. Firstly, due to the mechanisms of both asexual and sexual reproduction, early genetic material had to be able to self-replicate easily. For genetic material to be transferred between generations, the genetic information had to duplicate to be passed on. This is necessary for the transfer of genetic information. Secondly, early genetic material had to be able to catalyze simple cellular reactions. This allowed the original molecules of genetic information to serve many uses in the cell. Additionally, by having a catalytic function, the original molecules of genetic information ensured the expression of the genetic information. This was essential during the early stages of life on Earth since it ensured the survival of the organism. When considering both of these functions and the conditions of the early Earth, it is clear that RNA served as the precursor of biological heredity. When considering the structure of both acetyl-CoA and vitamin B12, the vestiges of an RNA-based world are visible in the presence of nucleotides in these molecules. Additionally, noticing how DNA nucleotides are formed from RNA nucleotides again reinforces this theory. Lastly, by analyzing the structure of ribosomes, it is clear that RNA subunits served as catalysts for protein formation. As a result, it is clear that RNA served as the foundation for the origin of hereditary material. Now, as a result of the creation and use of hereditary material, genetic variations were introduced into the reproduction of early organisms. These variations, which affected the growth and development of early Earth life forms, amassed over time to create vast genetic differences across multiple generations. Since these variations amassed over time, determining the origin of all species is important to understand the evolutionary process. Genetic evidence of common ancestry is based mainly on the study of DNA sequences of multiple organisms. By collecting data on the DNA sequences of a large amount of organisms, common segments of sequences can be determined. By knowing these common sequences, phylogenetic trees can be created to better understand when species of animals split from a common ancestor due to genetic mutations. Fossil evidence of common ancestry relies on the fossil record found around the world. Through carbon dating, fossils found in differing layers of rock formations have been studied to determine their age. Based on this preliminary analysis, a fossil history of life on Earth can be created to understand how changes in the life forms on Earth happened in chronological order. Based on this fossil record, it is clear that complexity in biological systems has increased over time. When considered together, both genetic and fossil evidence strongly reinforce the theory of a common ancestor. Using information gathered through both methods allows for an evolutionary map to be created that shows how different species have emerged throughout time. Although research in evolutionary biology has been successful, it has still not determined the sole common ancestor of all life forms on Earth. However, all evolutionary data still points to this unifying organism having existed. With both of these sources of evidence taken into consideration, it is clear that all of Earth's organisms shared a common ancestor. Genetic mutations in subsequent generations of this organism have resulted in the wide variety of life still found on the Earth today.